All right, welcome everyone to Scientists to Go. This is presented by Gulf of Maine Research Institute. And my name is Zoe. I'm one of the educators that works here in our lab. And today we're joined by Sam Bengs, who works in our Fisheries Lab. And she's going to talk to us a bit about the research that she does here to know more about food webs in the Gulf of Maine. And today, our presentation will be about 20 minutes long, where we'll listen to Sam and she'll have some activities for us to participate in. And then if you have any questions, you can feel free to throw those questions into the chat, and I'm happy to read them out to Sam at the end. We'll have about 15 to 20 minutes at the end for even more questions. So keep those in mind and throw them into the chat when you have them. But I'll go ahead and turn it over to Sam.
Great. Thank you so much, Zoe. I am going to go ahead and share my screen so we can all see some slides. How is that looking, Zoe? Okay. All right. Well, good afternoon, everyone. My name is Samantha Bengs. Everyone calls me Sam, though. And I'm a scientist here at the Gulf of Maine Research Institute. And I want to thank you all today for your time and your curiosity. So I'm really excited to share about my work with you all and give you an idea of what I do here at GMRI. There we go. So I wanted to share a quick roadmap for our time together today. We'll do a quick little intro on my path towards science, and I'll talk about the type of research I do here at GMRI, why studying creatures of all sizes is important, and we'll also talk about some cool new tools that my lab uses to study these aquatic species. And we'll have plenty of time, as Zoe said, at the end to chat about some comments and questions that come up while we're talking today. And I did want to mention I am going to have a group activity that we're going to work through together. So please feel free as we're going through our slides. Any questions you have, put them in the chat in real time. Zoe, if it seems really like appropriate to what we're talking about, feel free to ask those questions. And when we do get to the group exercise portion, Zoe's going to be a big help reading your answers as we work through this exercise together. So she'll monitor the chat for me. All right. Whoops. So just to give you all a little bit of a background on my path towards science, I've always loved animals and the ocean, but I did not grow up in Maine or even anywhere close to the sea. I grew up in Minnesota, so that's in the middle of the United States, and I loved my science classes as a student, and I was always curious, and I spent a lot of time outside playing and observing just how cool nature is. So I always loved science. But when I was a junior and senior in high school, I did want to go to college on the East Coast to study marine biology. But at that same time, I was really involved with band and orchestra, and I ended up getting a music scholarship. So, kind of despite what I wanted at the time, I wanted to come out east, but my parents said, you got a music scholarship, that's a good opportunity. So I stayed in the Midwest and took advantage of that scholarship for college. But it took me several years after studying music that I realized that I thought I could be happier if I chose and pursued a career path that put me back towards science. So I moved to Maine and went back to school for marine biology, and I ended up getting an internship here at GMRI while I was in school and kept in touch with the connections that I had made here. And about 2 years ago, I got a full-time position and have been able to work at GMRI as a researcher. So it's been great. So I think I have one of the coolest jobs at GMRI. I work in the Fisheries Ecology Lab. And what fisheries ecology means is that we study how fish populations interact with their environment and the other species that live in those environments. So I work closely with three other scientists and our boss. Our boss in a research job, they're called our principal investigator. So um, my team, we have the cool jobs. We get to be out on the water almost every day in the summer. And we go out. I'm actually going out tomorrow. It's going to be nice and cold, but we're going to be out offshore tomorrow. We work with a lot of different types of equipment, and we look at a lot of different types of aquatic critters, and we study where they are, um, what they're doing, what they're eating. Are they reproducing? Are they having babies? Um, are they healthy? We can ask a lot of questions when we regularly observe these animals where they live. So where the animals that we're studying live is obviously in the ocean, in bays, in rivers, near beaches. Um, we go all around the Gulf of Maine studying these different types of aquatic species. So I'm curious if anyone has an idea. If you look in this first photo here to the left, does anyone have an idea what I'm doing in that first photo? What is something really small, really, really small that we would catch in the ocean that we would need to kind of wash and strain into a collection bottle? Are there any ideas? Feel free to pop them in the chat of something really, really small that we would catch in the ocean.
Someone suggested maybe plankton or shrimp, algae.
Yes, plankton. Ding, 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 ding. Gold star. Um, Plankton are very small, but they are really important. So what I'm doing in that picture is washing plankton down into that collection bottle. That middle picture, we'll talk a little bit more about this in a few slides, but this is, I have filtered some water that we collected while we were out on the boat. And anything that's in that water is maintained. I'm not sure if my mouse is showing up. I gotta go on this screen. Sorry. Anything that's in that water is now maintained on that filter. And we'll talk more about that filter in a little bit. And in this picture on the right, this is me holding this is a small porbeagle shark. And this was caught in a net, and we're getting ready to put that right back into the water. But as you can see, research is really fun because you get to do a whole lot of different types of things, work with things really, really small, like plankton, all the way up to big things like a shark. So, getting really dirty here, but pretty clean here, working in a lab. So a lot of variety in the job of doing marine research. So, of course, working with sharks and fish is cool. But working with microscopic small plankton is also really rewarding because of how important they are. So I'm curious what you all think. Why do you think it's important to study a variety of species of all sizes, like little plankton all the way up to big sharks? Are there any ideas why we want to make sure we study all types of species?
Someone
Why would we?
suggested, they said we don't put things into the sea that will kill it.
Right.
Small things impact large things like sharks, and then they're all part of a food web, an ecosystem.
Exactly. This is I don't need to be here. This is such a smart audience. Um, in an ecosystem like the Gulf of Maine, all the species that live there together, they all work together and balance each other out. So let's think about our little plankton friends for a moment. These little guys are crucial because they form the base of marine food webs, and they provide about half of the world's oxygen through photosynthesis. And they also play a really important role in regulating the climate because they absorb carbon dioxide. So without our little plankton friends, the entire marine ecosystem would collapse. And that would affect not only ocean life, but also human food supplies and our global atmosphere. So it's really important to study the little things too. And if we think about our sharks, for example, while they are near the top of the food web, so not a lot of things can eat a shark. While sharks eat a lot of different fish and other prey items, they play a role in ecosystem dynamics as well. And another important thing that larger creatures do is they move energy around the ocean. And that energy first comes from our little plankton friends. So, like someone mentioned in the chat, everything is connected in these ecosystems, especially in marine ecosystems. So to get a little bit more deeper into this, let's talk about some food webs. I think we kind of already answered this. Um, but if anyone wants to take a stab at it, what do you think a food web means? If you had to define food web on a test question, what would you say a food web is? What is a food web?
Okay, we have a few answers. The movement of energy in a food chain, different things that creatures can eat. A food web is a representation of the feeding relationships between organisms.
Again, I don't need to be here. The scientists are already in the classrooms. You guys are so smart. So you nailed it. Perfect answers. So just think about all the food chains in an ecosystem. Think about everything that can eat something else. We can all eat multiple things, right? For example, you can have a banana for breakfast, maybe a cheeseburger for lunch, and maybe even ice cream for dinner. But whatever you are eating, you are getting energy for your body to function from that food that you eat. So from the sun to plants to all the foods that humans and animals eat, all of those energy sources from food are connected in a food web. So to kind of just illustrate this a little bit more and really understand how important each component of a food web is, we're going to do a little activity together. So first we're going to do a land-based food web, and then we'll follow that up with an ocean-based food web. And we're going to identify who would eat who on the next two slides. And then I'll act as our representative here. I will draw some lines between what we think. And remember, there may be more than one line coming from multiple things. So as we start this, just kind of give some, I'll go to the next slide so we can see it. We'll start with a land-based food web. Make sure to put in the chat kind of what you think goes from what to what, and Zoe will help us all read these answers. So let's start with this land-based food web diagram. What do you think on here? Where do you think the first source of energy is coming from? Any ideas? I might be kind of giving you a hint with my mouse.
Okay, a couple people have said the sun.
Okay, let me get a pen here. So we think maybe, what do you think is going to use energy from the sun? Maybe this tree? Can you see this pen on Zoom?
Yeah, we can see it.
Okay, great.
Yeah, we had someone say the grass as well, and
Okay,
even
great.
carrots.
And the carrots? Okay, good. So what's our next level? Who could eat grass, carrots, and benefit from this tree?
The rabbit or the bunny.
That would eat some grass, maybe some carrots too.
Humans will benefit from those. The chicken.
The chicken.
Hmm.
What is the chicken gonna go? To the grass?
Um,
Yeah. Okay, what else? Maybe what about our snake and our eagle friend? Where do we think they would go?
The eagle could eat the snake. That's one
Mm-hmm.
suggestion. The snake could eat the rabbit.
Yeah. Do you think the eagle could maybe eat the rabbit?
Someone said that the eagle could eat the chicken.
Eagle could eat the chicken. Yeah.
And yes, they do think, yeah, that the eagle could eat the rabbit as well. Um,
Okay.
and then the human would eat the chicken too.
Exactly. So as you can see, it's not just one straight line, is it? There's a lot of different lines going on. Now just imagine, what if we took out one of these pieces? Let's just say, for instance, we took out the grass. So all those little grass clusters would be gone. The balance would go completely out of whack and start to cause issues in each direction of this food web. So this is just one example of why it's so important to, when we're asking biological questions, we really need to ask about each component of a food web system like this. So let's move now. Let's go to a marine environment. What do you guys think? Do you think this starts in the same way as land, or is it maybe different? Where should we start this food web? What are we thinking? In a marine environment,
We have a couple people saying the sun would give energy to the seaweed, um,
right?
to the plankton.
Great. Very good. And that's another reason, again, why plankton are so important, because there are plants in the sea, but not like plants on land. So we really rely on our plankton friends to photosynthesize from the sun. So again, that's another reason why plankton are so important. They kind of act as the plants of the sea. So you got me. I tried to trick you and see if maybe a marine environment started differently, but it starts the same way. We all are really dependent on that energy from the sun and our organisms that can photosynthesize. They are the base of all the food webs. Okay, so we have our first step from sun to plankton and seaweed. What's next? Who's going to be eating plankton and seaweed?
The whales might be eating the plankton, the
Right.
crab as well, eating maybe the seaweed.
Mm-hmm. Very good.
Um,
Lobsters, when lobsters are smaller, they do also eat plankton. When they get a little bigger, they tend to start eating bigger things. They can eat some crabs. They can eat some fish that are slow. Um, I'm very proud of who said whale, because there are whales that exclusively eat plankton. So even a really big whale can survive with the energy that they get from little plankton. But that's not all whales. Not all whales eat plankton. So there are some whales that would probably be eating some fish. What else? Let's fill in a few more lines in this marine ecosystem. So we've got some fish. We have a bird up here. Um, who else would be eating who in this food web?
we did have someone say that some of the fish might eat plankton as well.
Right. So um, this isn't totally size representative. This is a smaller mackerel, and mackerel can eat smaller fish, but they can also eat plankton.
And then lots of folks saying that seagulls will eat fish and crabs.
Yes, very good.
Sharks will eat fish.
Oops. Sharks will eat fish. What about this guy? What about this cod? what is this cod gonna eat? So this is a relatively large fish.
One of our schools said that, yeah, big fish will eat smaller fish. So the cod might
Correct.
eat some smaller fish.
So this cod might be eating this mackerel. I think we've got a very good representation here of a marine food web. Does anyone see anything else that we may have missed? You guys are flying through this. But again, um, just think about for a moment, if we took out, let's say we took plankton. Let's say we took plankton out of this equation. This whole system would collapse. So this is why we want to study all the players in an ecosystem to understand how that whole system works together. And it's really important to watch out for when things might get out of balance so that we can help um, direct our research to study how to get things back to that balance where everything is working well together. So great job, everyone. Really impressed. I'm going to shift a little bit. So we've talked a lot about um, working on a boat, working out in the field on the water, and why we study those creatures. But I want to talk a little bit about another component of my job where I get to work in the lab. So it's another really cool component of my job. And Zoe, if you can for me, if we can show maybe Zoom hands or just some comments, how many of you students have spent some time in a science lab already? Are we getting to be in a lab setting?
Ooh, some people have lab every week. Yeah, lots of people have been in a science lab.
Awesome. So when I'm not on the boat collecting samples or doing research, I am in our molecular lab processing a lot of the samples that we collect. So in the lab environment, unlike the boat, I need to be very clean and very sterile and extremely precise with the chemicals and tools that I am using. So I really like that I have studied biology and science in multiple areas because that allows me to do both the work on the water and the work in the lab, which it's fun for me to have a lot of variety in the work that I do because even though the boat work is really fun, you don't necessarily want to be out on the boat all day every day, especially in the winter because it gets cold. So it's nice to have lab work to also be able to do, and having skills in a lab like this, you can really answer a lot more ecological questions with the research that we're doing here at GMRI. So, remember from a few slides ago when we talked about the small plankton and the big sharks. So we have to use very different equipment to physically catch those animals to be able to study them. Obviously, the plankton, we need something that's has a small enough mesh size so that the plankton don't swim through. And with sharks and fish and other animals, there's just a lot of equipment and time on the water to try and physically observe those animals. And that's what really informs our research. But there's kind of a new tool that we've been using that can help simplify things a little bit. If we simply just go out and take a sample of water where something is living, we can actually study them in the lab in the same way that we study them with catching them in a net, just by finding their DNA in the water. And so this can save a lot of time and effort when dealing with critters that can swim around a lot quicker than we can catch them. So I use a tool called eDNA or environmental DNA to study animals by finding their DNA signal in water samples. Has anyone heard of eDNA before or maybe even used it in any science classes? Just kind of want to gauge if have we heard of environmental DNA before?
I'm not seeing any responses yet. Someone said
Okay.
no, not yet.
Okay. So, um, the science behind environmental DNA, it actually stemmed from like forensic science. So like when you hear stories of, like how detectives solve crimes, how they do that is they use a really, really small amount of DNA that got left behind. Um, and they're able to take that really small amount and figure out who it belongs to because each of our DNA is unique to us and to each living species. So if all you had to solve a puzzle was DNA from 5 different animals, you could be able to piece together who it belongs to because it is different between each living species. So I think a really good analogy that I like to use to think about eDNA is think of it as a recipe. When you're using a recipe, it gives you the ingredients. So think of the components that make up eDNA are all the recipes of DNA. But the final dish depends on how you interpret the recipe and how you prepared the ingredients in your own way. So we each have we all use the same ingredients in our DNA, but it's how that final dish comes out, um, is what makes our DNA unique and different between between humans and between all the other living species. So because because DNA is so small and it lives inside our cells, right? So we've got millions of cells in our body, and that's where our DNA lives. It can be hard to imagine what eDNA kind of looks like in the environment. But let me ask everyone, how many of you have dogs and cats at home? Any pets at home? I would imagine some of our students have pets at home. Yes? Okay. Do they shed? Do you find their fur on your clothes, on your bed, on your furniture? Is there fur everywhere around your house? I know I have dog fur everywhere. Um,
Yeah,
that's
everyone's saying
eDNA.
definitely.
Your dog and cat fur around your home, that's eDNA. You can find your pet's DNA even if they weren't at home. It lingers. It's something that falls off of of a living organism as it's just going about its day. So that's kind of the concept of studying animals using eDNA is you don't have to actually see the animal. Even if I had my dog away from my house, if you went into my house, you could find eDNA of my dog, just like all of you can find that eDNA that's fallen off your pets at home. So it's just kind of a way that ecologists used some molecular techniques that were already being used to just frame some different questions that they wanted to ask. Um, so eDNA refers to DNA that is released from an organism or from an animal into the environment where it lives. So it's kind of gross sometimes, but if you really think about it in an ocean environment, it could be poop that came out of the animal. It could be mucus. It could be gametes, which refers to like the reproductive cells between male and female animals. It could be skin, hair. It could even be off of a dead animal, especially in the water. As if a fish died and sank, there's just DNA kind of coming off of that animal as it lays dead on the ocean floor. So how ecologists use eDNA is by taking a water sample and they can target a specific species. So like we all have our own DNA. If we wanted to go out into Casco Bay, take a water sample and say, hey, has there been a lobster in this water in the past few days? Um, we would take water, filter it back in the lab, and we use lab techniques we can isolate DNA from that water and clean it up. And we use a machine called qPCR. It's kind of this cool, I call it a really fancy microwave. It heats up and cools down in really rapid fashion. Um, and we put a lot of different chemistry into this qPCR machine that it can find that. So we're using lobster as an example. It could find that lobster DNA in our sample, and if it's there, it could make a really small amount into millions of copies. Um, but if we took water and there was no lobster signal in that water, it wouldn't make any copies. So this machine does a lot of work for us, telling us if something is either present or absent uh, from the water that we collected. So I know this map is kind of confusing to look at here. Oops. But I want to show you, if you can see these, we've got some blue dots here and some red dots. And this is some research that um, shows how scientists tracked two different, so the blue is a different species and the red is a different species of fish. And these scientists tracked these two species of fish just by using water. So they did not need to physically see these fish. They didn't put any nets in the water. They just collected water samples and took them back to the lab. And they were able to map out the presence of these fish all along the Chesapeake Bay area, just using their DNA signal from those water samples. So collecting water is a lot easier than putting nets into the water. So eDNA is a really exciting new tool for scientists who want to monitor animals that move around a lot. So we're really excited about what's possible if we spend a little bit more time collecting water rather than putting nets and spending a lot of energy with nets. Maybe we could study a lot more animals a lot more quickly just using water and focusing on that DNA signal. Now, I do want to mention that eDNA does have some downfalls. It's not a great tool for every single type of survey that you want to do. And think about it, because we are constantly shedding eDNA as we go about our days. As I've been sitting here talking, I've probably had some hair fall out, maybe like some skin cells. I've got Sam eDNA all over my desk, just as your classrooms are full of all your eDNA. Because eDNA is constantly falling off of us, it's very easy to contaminate your samples. So there is a lot of, you'd have to do a lot of cleaning, and you have to be extremely careful about not contaminating your samples. So, and another thing to think about is think about ocean water. There's a lot of waves, there's tides. The water can be very cold sometimes. It can also be warm. And the sun is always beating down on ocean water. This little image over here, you see, kind of pretend that this is eDNA falling off this fish. All of these components can cause DNA to degrade or fall apart a lot more quicker than a DNA signal could, say, maybe in soil or in the air. Because there's so many components in ocean water, we're still learning the best practices of is eDNA a good tool for maybe, say, trying to monitor a whale. That whale can swim a really long ways. So how do you know if water you collected, say, in the Gulf of Maine, did that water start in the Gulf of Maine, or did it come in on a wave from some other part of the Atlantic Ocean? So those are some of the components of using water sampling like eDNA, where we're still learning the best way to use this tool. But it is, like I mentioned, it's new and exciting, and we're thinking of the best ways of how we can utilize this new tool and its capabilities. So a big portion of the work that I'm doing here at GMRI is related to adding eDNA sampling to all of our existing surveys that we do around the Gulf of Maine, to see if we can see that DNA signal Um, is it strong enough to only use water? Or do we still need to use nets to catch the fish and physically say, oh, we see that we caught fish in the net? We'd love to get to the point where we can just use that water sample and rely on that DNA signal. So I hope that I know there may be some questions about DNA and how we're using eDNA in our work, but I hope that was a good kind of refresher on what we're doing here. And I want to thank you all for your eyes and ears and your participation as we were working through some of these questions. And I would really enjoy hearing some comments and questions from all of you. I don't know how we're doing on time. I feel like I may have gone over, Zoe. Sorry.
That's okay. Yeah, we have a bit of time for some questions. So if anyone has questions about eDNA or any of the work that Sam has done, or about food webs, feel free to throw them into the chat and I'm happy to read them out. Someone wants to know, what does your average day look like at work?
I'm going to give you two answers. In the summer, when the weather is nice, we're on the water a lot. So a typical day in the summer would be coming in, getting our gear ready, getting the boats ready, and going out and doing multiple types of surveys. Sometimes we take a little boat, sometimes we take a big boat. It kind of depends on the work because we have about 5 or 6 different big research projects that we're working on. So summertime, it's definitely out on the water most of the time. Right now, in the fall and winter, I come in and I do a lot of lab work. So if you remember that picture of the lab coat and everything looked very clean, that's where I spend the majority of my time. When we're not on the water, processing all the samples that we collect over those warmer months. And there is I'm not doing lab work constantly. There's a lot of writing and looking at data that I do at my desk, at my computer. But it's a good mix of being out in the field, in the lab, and at my desk.
Nice. I've gotten to go out with Sam on the boat a little bit in the summers, and it's always very fun. Um, and then we have a few more questions. So someone wants to know if it'd be possible for you to create a new species with the eDNA that you're collecting.
For GMRI's purposes, no. But there have been like we do not have the equipment nor the skill to create a new species. But something that's been really neat that other researchers have done, they have taken really old ice cores, so ice from parts of the world near the North Pole. They've been able to find, maybe some of you have heard that they have found woolly mammoth DNA that is an extinct species. They have found their DNA preserved in ice, and they've been able to make copies of that DNA and reproduce woolly mammoths. So while you can't create a new species, you can find the DNA of something that is extinct and potentially recreate that.
Very cool.
I want to ask, Jurassic Park is my favorite movie. And I would imagine if some of the kids have seen Jurassic Park or any of those movies, does it sound kind of familiar of using old DNA to bring something back to life?
Yeah, true. If you haven't seen Jurassic Park, it can be a good homework assignment.
Yes.
Um, okay, and then another question is, how can you tell if plankton are healthy?
That's a great question. Plankton are full of what makes them so nutritious to the things that eat them is they are full of lipids. So lipids are like a lot of fatty acids. And if a plankton, we collect plankton and we can do some analysis to see how much lipid they have inside of them. If it's under a certain amount, we can kind of think, ooh, they might not be in the healthiest of an environment. They're not getting enough nutrients into their body to make them send that energy up through the food web. So we look at that through their lipid content. We can also look, there's some plankton that have uh, types of shells around them. If there's shells or parts of their like carapaces are weaker, that kind of leads us to believe that they may not be the most healthy either. So that kind of depends on what nutrients are available for those little guys in their ecosystems.
A question about DNA. Just what does DNA stand for? Because we know that the E for eDNA is environmental.
Yeah, sorry if I breeze past that. DNA stands for deoxyribonucleic acid. So that's just a long term. So the D is from deoxyribo, N is nucleic, A is acid. It's just a fancy long name for the chemical components that make up DNA.
Nice. What degree is needed? We had a couple questions about your degree. So, what degree is needed for your job, or what degrees do you have?
I have a bachelor's degree in biology, and I am I'm completing a master's degree in marine biology. I'm defending my thesis next spring. So those are my degrees, and I have a bachelor's degree in music from a decade ago that is just kind of a bonus degree. But I studied biology, and then in my master's degree focused in marine biology. And I will say, just studying biology, that's where I got more of the molecular component of my studying, and that really helped me to be able to do the lab work portion of my job.
Very cool. Um, One question is, have you ever been in a dangerous situation at work?
Yes, I have. We've been when we go out on the boat, sometimes we can be pretty far away from land, and we've had some mechanical issues on the boat. Um, I have had a hook go through my hand. That wasn't very fun. Um, I have I did get a t tail slapped by a shark once. That was fun, but also a little scary, and I had a good scar from that. But it's all part of the job, and we're surrounded with really good staff, and we do a lot of training that makes us prepared for when things go wrong, because it is kind of inevitable when you're working in a somewhat dangerous field that things can go wrong, but it's all about the training that you have to be able to um, deal with those issues and make sure that everyone is safe out on the water. So I have had some issues, but everything has always turned out because we've had such good training around us to prepare us for those situations.
Speaking of training, another question was, do you need to be trained in deep sea diving for your research?
No, I have actually had zero. I've never scuba dived. I have no scuba experience. Um, that's pretty, I think a lot of people think marine biologists have to um, be able to scuba dive. Um, you don't have to. Actually, a lot of places, because that is such a risky activity. Um, this where I went to school, they didn't have insurance to cover any researchers doing their own scientific diving. So a lot of that is actually hired out to scientific divers who have the insurance to cover that high risk activity. Something that has helped me in my career is I got my captain's license. So I learned how to be a boat captain, and that has helped me be competitive in the job market for being a marine biologist. But you don't have to scuba dive. I know plenty of people who do, and I'm jealous, and I do at some point want to learn how to scuba dive, but it's not necessary for my career.
Cool. Um, and a question about just the process of your research. How long does it take to find the DNA of an animal?
So, takes about we collect the water, we filter that water in the same day because remember how I mentioned that DNA can degrade, it can fall apart quickly. So as soon as we collect that water, we get it on ice and then we get it back into the lab and we filter it down. And that filter that can then sit in a really cold freezer, -80 degrees Celsius. So that is really, really cold. And once we put that filter in that freezer, it can stay there for eternity. It's archival level preservation. And then so what really happens is in the summertime, I filter a lot of water and then those filters sit in the freezer. And then what I'm doing now is since we're not on the boat as much, I'm in the lab processing these filters. Um, so in a perfect world, if I could do all the work just um, process after process, I could probably collect water and maybe at the end of the next day be able to determine whose DNA was in that water. But just based on the nature of the job, it's not really possible to do it that quickly, just because I have to um, get back out on the water to utilize that good weather during field season, if that makes sense.
Mhm. Cool. A couple of quick questions just about animals that you've seen doing your research. Well, I guess one is about animals that you've seen. So, what is the most unusual animal that you've ever found?
Mm. I this summer we saw a basking shark. So it was probably 25 to 30 feet long. It was huge and it got right next to the boat. So that was pretty exciting. I've seen a thresher shark, which is the shark with a really big long tail, and it had breached out of the water, and we saw its tail flipping. Um, that was really neat. Um, a couple weeks ago in the lab, we collect fish and we look at what they are eating. So we had opened up the stomach of a pollock, and there was a puffer fish inside of a
Whoa.
pollock stomach. And we don't typically see puffer fish in the Gulf of Maine. So that was a pretty exciting find. Um, and there have been a few times where we have found some lobsters that are cool colors. I've seen a blue lobster. I've seen a white and yellow lobster. It looked like a banana split. Uh, so yeah, there's always something unique to see in the ocean. No matter how many times you go out, every single boat trip is different, and you see different things every day.
Nice. And then I'll just do one final question. What is the biggest highlight of your career so far?
Oh, that's a
Yeah, it's a
good
hard.
question. That's it. Kind of similar to what I just said, like every day is different. Um, and it's just when you think you kind of like have the ocean figured out, it surprises you. Um, so it's really a highlight for me to have spent enough time on the water now that I realize that we can never learn enough about the ocean because it's always changing, it's always different, and it's never going to be the same trip. So I think being privileged enough to spend that much time on the water and really having a deep respect for the ocean has been a highlight for me because I think a lot of people love the ocean because it's so beautiful to look at and we know the fun creatures that live in it, but to be able to work on the ocean day in and day out has truly been an honor, and I'm really really happy that I get to do it.
Well, thank you so much for talking to us about your research. And thank you everyone for joining. This was a really fascinating presentation. I just have a quick plug for anyone that's interested in getting involved a little bit more with our work here at Gulf of Maine Research Institute. If your classroom's interested in doing science and engaging a little more, we have a student science journal called Findings from the Field, as well as an in-person symposium that happens in March. And we'd love to have any and all of you participate. So I'm going to post a link to how you can do that in the chat, and I'll also send it in our follow-up email. But thank you again to Sam and to everyone for joining. We hope to see you at a future Scientist to Go presentation.
Thanks, everyone.